Ferro, and so that we can start off with the technical sessions. Thank you very much.
Thank you, Professor Anil Mishra. He is in our faculty of the department and also the technical director of this e-seminar. So he would be the right person to say when these things what is told to you on the technical perspective. Now for the day 1, June 25 today, the first speaker is Professor Vito Ferro, the Deputy Director, Biotechnology Program, School of Chemistry and Molecular Biosciences, Australian Infectious Disease Research Centre, the University of Queensland, Brisbane, Australia. Professor Ferro research interests are in carbohydrate chemistry and medicinal chemistry, with a focus on the synthesis of compounds to probe and or inhibit carbohydrate protein interactions involved in disease processes. Previous work in this area resulted in the discovery of a potent inhibitor of angiogenesis and metastasis that is currently in phase 1 clinical trial in cancer patients. Current research interests are the development of artificial anticoagulant as replacement for heparin and LMWH, heparan sulfate, mimetics as antiviral agents, and pharmacological chaperone therapy for lysosomal storage diseases. This is a brief introduction about it. So I would request Professor Vito Ferro to please start your lecture today. Thank you.
Thank you, Professor Kare. Can everyone hear me? Do I need to increase the volume, or is that okay?
No, it's okay. You can hear it.
Perfect. Excellent. Okay, so we'll go into share screen mode and begin my presentation. Okay. All right. Hopefully you can all see that now and ready to go. So firstly, I would like to thank Professor Naveen Kare and the organisers for the kind invitation to this wonderful e-symposium. And I guess in the current climate where it's difficult to travel, this is the way that things will need to be, and it's fantastic to be part of it. And I'm very honoured to be the first speaker as well, although perhaps that has something to do with being in Australia, where we are. It's 4 p.m. in the afternoon here, and we're ahead of everyone. But nonetheless, it's still a great honour. Okay, so today I'm going to talk about heparan sulfate mimetics as antiviral agents for COVID-19. Okay, and I guess you'd have to be living under a rock to not know about the current coronavirus pandemic, COVID-19. And I just pulled out some figures yesterday from the WHO website, and as of yesterday, we'd hit 9 million cases worldwide, and we're closing in on 500,000 deaths. And I haven't seen today's numbers, but they're no doubt much higher. And looking at this graph, which is the from the WHO showing the cases, then I think it's pretty clear that we're in a bit of trouble with this pandemic, and we really need vaccines and therapeutics ASAP. Okay, now just going to. Okay, so in terms of COVID-19, just a moment, I've got this. There we are. Okay, I just had something from Zoom obscuring my seminar. Can I move this somewhere? Okay, all right. In terms of COVID-19, it's quite a nasty disease, but for some of us, or some patients, they don't even know they have it, and it can be quite mild. But for others, it can be quite problematic, and it comes in three pathological phases: an early infection phase, where you basically either don't know you have it or you have mild cold or flu-like symptoms. It then can move to a pulmonary phase, and then ultimately onto a severe inflammatory phase, or the so-called cytokine storm, where your immune system basically goes into overload, and that leads to the problems and ultimately the deaths that we see. And unlike other coronaviruses, the SARS-CoV-2 virus can infect a range of different types of cells, not just those in the respiratory tract. And it can cause viral pneumonia, which leads to the acute respiratory distress syndrome, or ARDS, as it's called. It also can cause pulmonary coagulopathy, which is basically blood clots forming in the lungs, which then can lead to respiratory failure. And it can affect many other organs and result in multi-organ failure, etc., and then ultimately death. So very nasty disease. Okay, so here is a picture that I found on the internet of the coronavirus. You've probably all seen one like this. The coronavirus looks basically like a golf ball with these spikes on them. And this spike, or spike protein as it's known, is a glycoprotein. So it's heavily glycosylated. And that's another story in itself, and speaks to the importance of glycosylation in the pathology of viruses, including the coronavirus. But there's another carbohydrate that is also very important in the infection of coronavirus, and that is heparan sulfate. And heparan sulfate is a polysaccharide that's found on the surface of almost every mammalian cell, and many viruses, including coronavirus, use it as an entry receptor or co-receptor. So in fact, the coronavirus, for example, will initially attach to the cell via making an attachment between the spike protein and the heparan sulfate. And this results in the initial attachment to the cell, and it can then engage with its receptor, which is called ACE2. And in the SARS-CoV-2, there's also another protein involved in that process, which then leads to internalization and infection. And indeed, it's been shown by my collaborators and others that SARS-CoV-2 spike protein indeed interacts with heparan sulfate. And in this COVID-19 world, where there's urgency to report research findings, many of the reports that we're seeing in this area are on these preprint servers such as BioRxiv. Okay, so heparan sulfate. So what is it? Well, it's a linear long-chain polysaccharide, which is found typically attached to a protein core to form a structure called the proteoglycan, and that's found on the surface of every mammalian cell, as I said. Now, structurally, there's a linker sequence that links the polysaccharide to the protein, but then it's made up of these repeating disaccharide subunits, which are made up of a uronic acid, a glucuronic acid, or iuronic acid linked to glucosamine. And there are sequences or domains within these heparan sulfate chains that are made up mostly of this non-sulfated disaccharide sequence on the left. And then there are domains that are highly sulfated, and they contain a high proportion of iuronic acid and a high level of sulfation. And these are very diverse and heterogeneous structures. And most of the binding to target proteins happens at these S domains, these highly sulfated domains. And this will be the case with the SARS-CoV-2 spike protein. Now, a related polysaccharide I should mention now is heparin. Okay, so heparin can be thought of as an oversulfated version of heparan sulfate. Okay, so it's a very similar polysaccharide, but it has a much higher proportion of iuronic acid and a much higher proportion of sulfation and variable sequences. So it can be thought of those sulfated domains of heparan sulfate joined together. And heparin is an anticoagulant drug that's been used for decades, and it's readily available from animal intestines, usually pigs. And because of its ready availability and because it's very similar to heparan sulfate, it's commonly used as a proxy for studying heparan sulfate protein interactions. So indeed, any protein that binds heparan sulfate also binds heparin. And so heparin has also been used as a means to block heparan sulfate protein interactions, and this includes in viruses. And so heparin is actually known to block infection of viruses by preventing that initial attachment of the virus to the cell. Okay. All right, and so recently there's been a whole range of studies from a number of labs, and these have appeared mostly on the preprint servers, and essentially all of these are showing that heparin or low molecular weight heparins can effectively interact with the spike protein of SARS-CoV-2 and also block binding to cells. And also interesting as well, there's been a report as well about the benefits of heparin for mitigating the cytokine storm in late stage COVID-19 patients. And so heparin has in fact, or is in fact being used clinically at the moment during the pandemic in a number of settings with the late stage patients, either to prevent or to treat the coagulopathy in the lungs, but also it's having some effect on mitigating the cytokine storm, and indeed, in some cases unknowingly to the physicians, may also be having some effect on the virus infectivity and transmission as well. So heparin is very interesting, and it's currently in one form or other is in clinical trials in a number of places. I know, for example, at the moment in the United Kingdom, heparin is one of a number of drugs that is in clinical trials for COVID-19. Okay, so this now leads me to a story about what we've been doing in this area, and this is a story about a heparan sulfate mimetic known as Pixatimod, or also known as PG545 and sometimes ZU545. Now, this is a clinical stage heparan sulfate mimetic being developed by a company in Australia called Sukero Therapeutics. And in fact, before I joined the University of Queensland, I worked for what was then called Progen Pharmaceuticals in the drug discovery area there, and I was the director of drug discovery and led the medicinal chemistry team that developed this compound, and ultimately went into clinical trials, and it's in development at the moment. So just quickly say something about the structure. So it's a heparan sulfate mimetic. So it has this tetrasaccharide portion which is fully sulfated. Now this tetrasaccharide derives from maltotetraose, okay, so not from heparan sulfate, and of course it's much easier to synthesize because it's not a heparan sulfate sequence, which are themselves quite challenging to synthesize. This is quite simple to synthesize. Essentially, you take maltotetraose, protect it, convert it into a glycosyl donor, glycosylate with this steroid cholestanol, and then you deprotect and fully sulfate. And synthetic details are described in the JMed Chem paper here. Okay, so this is the compound. It has potent anti-cancer properties, anti-metastatic and anti-angiogenic properties, as Naveen mentioned at the start, and anti-inflammatory properties. It's one of the most potent inhibitors of heparinase that is known. Heparinase is the glycosidase that degrades heparan sulfate extracellularly and is involved in many aspects of cancer progression and inflammation. It's also a potent inhibitor of interleukin-6, which is one of these key cytokines involved in the cytokine storm associated with COVID-19. And heparinase and IL-6 are actually connected in some way as well, because higher levels of heparinase result in higher levels of IL-6 as well. And in addition to those properties, Pixatimod also has immunomodulatory activity, which I won't go into, but it's an important aspect of its anti-cancer properties. And so this was in a phase 1A clinical trial, and at the moment, as we speak, it's in a phase 1B trial in cancer patients with pancreatic cancer and bowel cancer in combination with a drug called Opdivo, which is an immune checkpoint inhibitor. And so far it's been safely administered to approximately 80 patients. And in the cancer trials, the safety profile has been quite okay, quite tolerable. And there's been some evidence of disease control. And a couple of other interesting factors or features of Pixatimod are that it has a very long half-life. And this is due to the steroid side chain, which was introduced initially into this class of compounds in order to increase the half-life. That was the initial rationale for doing that, and it worked beautifully. So this is dosed only once a week as an infusion. Another aspect about the steroid part is that it reduces the anticoagulant activity of the compound. So it has only mild anticoagulant activity. Now, I mentioned a slide or two ago about heparin and how it's being trialled and it has some beneficial effects in COVID patients. Unfortunately, heparin is a potent anticoagulant, and so that sort of limits the doses that you can give in order to get a beneficial antiviral and anti-inflammatory effect versus causing bleeding. So that's an issue. So this is quite a advantage of Pixatimod. Another advantage, of course, is that it's a single chemical entity, single compound, whereas heparin and derivatives of heparin are all heterogeneous mixtures. Okay, now the other important thing about Pixatimod is, there we are, is that it has broad spectrum antiviral activity. Okay, and this has been known for quite some time, and we've done quite a few studies of this over the years. And here, this slide, I just present some of this data to you. So up the top here, we have a table showing some, not all of the different viruses that have been tested against, or for which Pixatimod has been tested. And you can see here, it's shown antiviral activity against HIV, virus that causes AIDS, respiratory syncytial virus, herpes simplex, Ross River virus, and dengue. And the numbers here, the EC50s, are quite good, some into the nanomolar, in the sub-microgram per mil range, so quite potent. And here's some recent data. So down the bottom here is some of this data showing Pixatimod, or PG545, here in the blue, against dengue. So it has quite a potent EC50 here. And this graph here is just showing that it is non-cytotoxic, and certainly, or not cytotoxic at concentrations that are therapeutically relevant. Now, over here on the right is some other data also showing its effects in reducing IL-6 in the setting of an antiviral disease. So on the panel on the left here that I'm pointing to is showing how PG545, also known as ZU545, inhibits IL-6 induction in cells that's caused by the dengue virus non-structural protein 1. And that's quite a potent effect here. And the one on the right is showing a reduction in IL-6 expression in the joints of mice infected with Ross River virus. Okay, and all of these studies on the antiviral activity have all been published, and some of the references are there if you're interested. Seems to be taking a few clicks to change. Okay, all right. In addition to the antiviral activity, which is essentially due to blocking of the virus attaching to the cell and infecting the cell via blocking the interaction with heparan sulfate. In addition to this activity, Pixatimod has a very unique activity not found in heparin or any other type of heparan sulfate mimetic, only in this particular class of highly lipophilic heparan sulfate mimetic, and that is virucidal activity. So what does that mean? That means that the compound can actually inactivate or kill the virus, if you like. And this is shown here in the setting of herpes simplex 2. So here's a control virus, and here are these are electron micrographs. So here's a virus that has been disrupted by the Pixatimod. So essentially, we believe that the steroid portion of the molecule interacts with the viral lipid envelope and disrupts the virus, causing inactivation. And so this is quite a unique feature. So not only will it block infectivity, but it will also kill the virus, which is very useful. In addition, it has been shown in 3 models so far of virus disease in animals that it has efficacy. So this has been tested in the the prophylactic HSV2 genital infection model, in the prophylactic Ross River virus model, and in a therapeutic dengue model. And the references are all here. This has all been published. And here is just one or some of the data this is in the HSV2 model, showing a protecting effect of prophylactic administration of Pixatimod in this disease. Okay, there's a time lag waiting for my slides to change, but here we go. All right. So to summarize, we have this heparan sulfate mimetic, and it has all these useful properties. So it inhibits heparinase and IL-6, which would be important for the cytokine storm affected patients with COVID-19. It's been administered to patients already. It has a good safety profile. At least 80 patients have had this drug. It has a long half-life, so it only needs to be administered once a week. And it has low anticoagulant activity, which is good. And I've just shown you it also possesses broad-spectrum antiviral activity. So this is great. So this sounds like a perfect drug repurposing compound. This sounds like we should be testing it in COVID-19 or SARS-CoV-2. And so that is the only question left, and that is, does it work against COVID-19? What about SARS-CoV-2? Okay, and so the question, and the answer to the question is yes, it does work against the virus. So far, we have done a number of studies, and the first of these is to look at the interaction of the drug with the spike protein receptor binding domain of the virus. And these data are showing you some of those interactions. So panels A and B here is just the same figure in A, just blown up. This is the circular dichroism spectra of the SARS-CoV-2 S1 receptor binding domain alone, which is in black, and then with heparin in red and Pixatimod in blue. So CD, circular dichroism, or CD spectra, is a means to be able to look at the secondary structure of a protein. And when something binds to a protein, if it induces a conformational change, then we can see that in the CD spectra. And so this is evident in these spectra because we can see that the blue and the red lines are well separated from the black line. And then you can also analyze it to determine the amounts of changes or the types of changes in the secondary structure. And then the panel D at the bottom here, then panel D here at the bottom is some data from a competitive ELISA assay where we're looking at the binding of heparin and Pixatimod in either black or grey to the S1 RBD, and we see indeed that it does bind. In this case, heparin does bind more strongly than Pixatimod, and that's to be expected, I guess, given the size of heparin relative to Pixatimod. Okay, so next we did some molecular dynamics simulations. And so fortunately the crystal structures of the S1 protein has been solved by a number of groups. And so we took a crystal structure of the S1 protein together with the ACE2 receptor, and we did a molecular dynamics simulation with Pixatimod. And we did what we call a blind molecular dynamic simulation. And that is, we didn't tell the system beforehand where to bind. So often when you're doing these simulations, you know roughly where the active site is. And so you define a box around that active site and you throw the ligand in there and you say, okay, find a happy place to bind within that box. But in this particular case, we didn't define any active site. We just threw the molecule in there, or the molecules in there, and told them to find each other, as it were. So these, naturally enough, are very computationally expensive simulations that take several days on the supercomputer. But in the end, we were able to get some interesting data. So here on the left is from the simulation. This is Pixatimod here binding to the S1 protein. Now, what it does is the sulfated tetrasaccharide part of Pixatimod here binds to a part of the heparan sulfate binding site. Heparan sulfate binding site is here. And in other simulations, and I'll show you in a moment, heparan sulfate does bind across here. All these blue residues here are all positively charged amino acids that will make an interaction with the sulfates on heparin or heparan sulfate. But Pixatimod only attaches itself partly to the heparan sulfate binding site, but then the lipophilic side chain also interacts with this so-called loop region of the spike protein. And if we overlay the crystal structure with the structure from the simulation, we've taken out the PG545 for clarity, and if we look at these two regions, we can actually see that there's been a significant conformational change in the protein, and this is actually in complete agreement with the CD spectra. So our simulations are also suggesting that PG545 or Pixatimod should bind to this protein and induce a conformational change. And indeed, in this area here, it's suggesting that it can not only block the heparan sulfate binding, but it can also interfere with the interactions with the ACE2 receptor, which is the main receptor for the virus. Okay, and just to show you, this is another view of the same spike protein receptor binding domain, but this is a simulation with a heparin, I think it's an octosaccharide, and it just binds here in the heparin binding site, whereas in the previous figure you saw Pixatimod binding in this part of the heparan sulfate binding part and interacting with the loop region over here. So it has an interesting way of binding to the protein, and suggests a couple of different ways that it might be able to block infectivity by the virus. Okay, now just quickly, here's another view from the simulations showing Pixatimod. On the left, we're showing the Coulombic surface, so the blue regions are positively charged, the red regions negatively charged, and we see here how the sulfated tetrasaccharide portion is interacting with the blue positively charged heparan sulfate binding region. And then over here in this panel, we've changed to change the colours to show where we have hydrophobic patches, so that the orange here is a hydrophobic patch, and that's showing how the steroid is interacting with that hydrophobic patch, and that's all in complete agreement with what we saw earlier. And so we've shown that it binds to the S1 protein. Our modelling also suggests that it should do that, and here are some of the data that we now have showing inhibition of infectivity or antiviral activity, if you like, of Pixatimod against SARS-CoV-2 virus isolates. And these are three separate figures done all slightly differently in 3 different laboratories. The first one in the UK, the one in the middle in Sweden, and the one on the right here in Brisbane. So using different isolates, and the data here is summarised in the table at the bottom here. Okay, and so we are showing here EC50s of around or anything from just under a micromole, a microgram per mil, up to say 12 or so micrograms per mil. So important things to note here are the CC50. So this is the cytotoxic concentration. So the concentration at which Pixatimod kills the cells. So in fact, we can use this to work out the selectivity index. So in other words, these figures here showing that the concentrations required to kill 50% of the virus, or to inhibit 50% of the virus, are 20 to 30 times lower than the cytotoxic concentration. So that's a good thing. The other important point to note with these numbers is the Cmax of Pixatimod. Now, the Cmax is the maximum plasma concentration in patients who have been dosed the recommended dose of 100 milligrams of Pixatimod, and that figure is approximately 30 micrograms per mil. So in other words, the EC50s here are much lower than a maximum plasma concentration that we see when we dose the drug. So that's a very good thing, because there's been some various other studies done with other drugs where there is certainly an antiviral effect, but the Cmax, the maximum plasma concentration achievable in plasma, is much less than that required to have an antiviral effect. So that's not going to be an effective drug. Whereas this is saying that the EC50 is well within the therapeutic range. So that's really exciting. Okay, so this slide then just summarizes the...
please conclude, because the time schedule, the next speaker is waiting actually.
Yes, okay, this is basically the second last slide. So this is the third last slide. So this is just summarizing the different mechanisms of action of the drug. And so that is to basically block the attachment to the cells and prevent infection, potentially to have virucidal activity to inactivate the virus, although experiments are still ongoing for that, and then inhibition of IL-6 and inhibition of viral spread via inhibition of heparinase, which is involved both with IL-6, and also it's been shown to be involved in viruses escaping from a cell and infecting the next one. So what's next? All this work that I've talked about has just been submitted to the preprint server actually last night, so it should be available in the next day or two. What's coming next are more in vitro studies. We'll be moving on to in vivo studies in a number of different models, the mouse, the ferret and the monkey, and Zucchero Therapeutics are aiming to go into a phase 2 clinical trial by the end of the year in SARS-CoV-2. And while that's happening, we're also looking to design and synthesize new heparan sulfate mimetics that are specifically optimized for SARS-CoV-2 rather than Pixatimod, which was originally an anti-cancer drug and being repurposed. Okay. And so finally, I would just like to acknowledge all these people who worked on it. So you can see there's a multinational team from Queensland, from the United Kingdom, and from Sweden, and supported also by Zucchero Therapeutics. And I'd like to acknowledge also the Australian Infectious Diseases Research Centre. And thank you for your attention. I'm sorry I went a little bit over time, but I'll blame my slides, which for some reason on Zoom were changing rather slowly. But that's it from me, so thank you very much.